Hi, I'm Rachel Connor, a senior in the Medical Laboratory Science Bachelor's Degree program at the Texas Tech University Health Sciences Center in Lubbock, Texas. I've had the opportunity to be involved in a research project geared towards trying to determine if the bacterial causes of sepsis remain in cadavers after the embalming process is completed, along with Mr. Johnson, Mr. Jones, Mrs. Veris, Dr. Gilbert, and Dr. Bashir. Cadavers are extremely important in the medical field because they allow future medical doctors, researchers, and professionals to develop proficiency on identifying human anatomical structures, as well as enabling medical professionals to perform research that improves healthcare. Cadavers have been labeled as irreplaceable learning tools by other researchers due to their enormous impact on teaching future medical professionals. Willed bodies are in short supply, and unfortunately, the TTU-HSC Anatomy Institute loses 12 to 20 of these invaluable resources annually due to sepsis on the medical chart or death certificate. Sepsis is a condition of having microorganisms in the blood, and it raises the concern that perhaps the bacterial causes of sepsis might remain in the body post-mortem, and that the body could pose an infectious threat to those who are exposed to it. The process of embalming fixes protein, and its goal is to preserve and sanitize the body after death. The purpose of this research study is to find out if the bacterial causes of sepsis remain after embalming through culturing arterial blood samples collected within 5 days post-mortem and comparing them to samples taken after the embalming was done. In this research, all bodies donated to the TTU-HSC Willed Body Program were included regardless of sepsis association. To obtain the arterial blood samples, sterile procedures were used to collect 20 milliliters of arterial blood from approximately 5 days post-mortem cadavers. Then, 10 milliliters of that sample were transferred into a BD Bactec aerobic blood culture bottle, and the remaining 10 milliliters were transferred to an anaerobic BD Bactec blood culture bottle. Once the blood culture bottles were filled, they were immediately transported to the MLS training lab to be placed in the Bactec 9050 for incubation and continuous monitoring. The Bactec alerts to changes in the CO2 levels, which is indicative of growth. All blood culture bottles were incubated for at least 5 days to ensure that slow-growing bacteria would be detected. If the Bactec alerted that there was growth, small samples were taken using a sterile airway needle and set to culture plates and a gram stain. A positive aerobic culture bottle is set to a BAP plate at 37 degrees Celsius in ambient air and a chocolate plate at 37 degrees Celsius in capnophilic air. A positive anaerobic blood culture bottle is set to a CDC ANA sheep's blood agar plate in an anaerobic chamber at 37 degrees Celsius and a chocolate plate at 37 degrees Celsius in capnophilic air. Growth on the plates is assessed at 24 hours. Normally, the MALDI-TOF was used to identify the bacteria that grew out on the plates, but if that was unsuccessful, an API manual identification system was used. After the embalming procedure is completed, the blood collection and incubation processes were repeated. During this study, samples from 20 cadavers were processed, so altogether there were 80 blood culture bottles. 17 out of the 20 cadavers had growth observed before embalming in at least one of their blood culture bottles, and 31 of 40 blood culture bottles collected prior to embalming had bacterial growth. The graph titled Frequency of Genus Identification shows the number of times that a certain genus of bacteria was found in the pre-embalming blood culture bottles. And the table titled Pre-Embalming Predominant Isolates shows the bacterial genus and species of the microbes found in the blood culture bottle or bottles from each cadaver that showed growth. None of the 40 post-embalming blood culture bottles showed any sign of growth. Willed body donors are essential in the advancement of medical education, research, and healthcare. In addition to providing a contribution to medicine, for some, willed body donation offers the chance to leave a lasting legacy through contributing to the progress of medical understanding and the enhancement of healthcare for prospective generations. Having to reject willed bodies due to sepsis on the medical chart or death record is detrimental in so many ways, and there is responsibility to investigate every conceivable possibility to ensure these irreplaceable resources are not wasted. The results of this study indicate that embalming mitigates the bacterial infection risk in septic bodies since there was never any bacterial growth in the post-embalming samples. Past studies show a reduction of microbial growth after embalming, but not complete elimination. This discrepancy between the data from this research study and others might be due to possible differences in the formulation of embalming fluids and differing sample collection. A limitation during this research is that only one sample type was studied. Further research must be done with different specimen types, such as bodily tissues, to demonstrate that the bacterial causes of sepsis cannot live on after the embalming process. However, the data gathered by this experiment suggests that the embalming procedure is capable of removing microorganisms from the arterial blood of cadavers, possibly making the body non-infectious. These are the references that were used in the making of this research poster. Thank you so much for listening.